Hello and welcome to Lecture 9. Today we will talk about capturing and using energy. And with this lecture, we basically start a new chapter in our class. So we're going to go talk about something quite different from what we discussed before. Okay, so what is it that we're going to talk about? Well, we're going to see how energy is obtained, what energy is, what kind of molecules contain energy, how they can contain energy in these molecules. And then we're going to see how metabolism, so that means the breakdown or the change of molecules within an organism, can allow an organism to get the energy that it needs to perform all the functions of life. So your book has a nice overview of terms. So basically what this figure shows here is that you can classify organisms in a variety of ways depending on how they get their energy. There are the phototrophs, so those are the ones that make their own, that get their energy from the sunlight. And then there are the chemotrophs that get them from a chemical compound. And then there are heterotrophs and autotrophs. So you have autotrophs and heterotrophs here that are also phototrophs, and then chemoautotrophs and heterotrophs. And basically what that means is that an autotroph gets its energy from carbon dioxide or another inorganic source, while the heterotrophs use carbon that's previously incorporated into a compound, an organic compound, by a different organism. So they're not making it straight from the surrounding, from the environment, from inorganic sources. And so a plant, of course, is a great example for a photoautotroph, while an animal is a great example for a chemoheterotroph, right? It uses chemicals for getting its energy, sugars. This is a little sugar molecule here. And that is the carbon in that sugar molecule comes from some other organism that made it. In many cases, it's a plant that made that sugar that an animal then eats. But there's a number of crossover cases, especially when it comes to algae and bacteria that do different things. So we are going to take a look at how energy is converted and what energy really is with respect to life and organisms using that energy. So there's the term metabolism that I just used, and basically that means energy conversion. So it's the energy conversion within molecules within an organism, and as those molecules change in their form, as they basically undergo reactions, they can either be broken down or they can be built up. And so there are two different terms for that. There's catabolism, which is the breakdown of molecules, and usually that produces ATP. So here you see macromolecules such as carbohydrates, proteins, fats, and nucleic acids, which we discussed in the first section of this class. And in many cases, when you break those down, you can produce energy in the form of ATP. And we've talked about ATP just a little bit, but we haven't really discussed exactly what it is yet, and that will happen in this lecture. And then the subunits, as we're putting those together to produce macromolecules, that often costs ATP. And so when you have to invest ATP in order to make these larger molecules, then that is costing the ATP, and that is called anabolism. So catabolism, breakdown; anabolism, build up, costing versus producing ATP. And both of those together, the entire circle here, that is metabolism. Okay, so let's take a look at energy in general. So how does a body or a substance or a molecule contain energy, and how is energy converted? Your book talks a little bit about thermodynamics and the laws of thermodynamics, and you can take a look at those. Those are not going to be a big part of this class, but we do want to understand what energy is and how it is converted, because specifically when it comes to chemical energy, we want to understand where the energy comes from that we need for metabolism. So if you look at this cartoon here with this ball at the top of the stairs, this ball has high potential energy. That means it contains energy that it could use. Right now it's not using it, it's just sitting there. But it could use it, and so it has a lot of potential. It has a lot of potential energy. You just give it a little bit of a tip, and then it will start to bump down the stairs. Because it has the energy, it can just go roll down by itself. And that energy, as it's rolling, as it's moving, that is called kinetic energy that it's using. And then at the end, you can still ask how much potential energy is in this ball, and you'll say there's much less in there now than there was before, because now it has used its potential energy in the form of kinetic energy, and now it only contains low energy. So potential energy is stored energy, kinetic energy is energy of motion. And if you think about potential energy not in terms of a ball sitting on top of a stairs, but maybe water behind a dam or energy stored in a food molecule, those are all examples of potential energy. And then we also have chemical energy, which is potential energy stored in molecules, in chemical molecules. And this part here, the chemical energy, that is what we will spend quite a bit of time on finding out more about. So let me show you a little video that your book provides.
The energy of a system is its capacity to do work. For a cell, energy is needed for such processes as synthesizing DNA, RNA, and proteins, pumping substances into or out of cells, and cell division. There are two forms of energy: kinetic energy and potential energy. Kinetic energy is the energy of motion. A ball bouncing down a set of stairs possesses kinetic energy. Kinetic energy is associated with any kind of movement, whether a person running or a muscle contracting. Similarly, light is associated with the movement of photons, electricity with the movement of electrons, and thermal energy with the movement of molecules. So these are all forms of kinetic energy. By contrast, potential energy is stored energy. It depends on the position of an object relative to its surroundings or on the object's structure. A ball at the top of a set of stairs has a large amount of potential energy. Given the chance, the ball will roll down the stairs. In this case, the potential energy of the ball is converted to kinetic energy. A dam in a river also stores potential energy. If a hole is introduced in the dam, the water flows through it. Similarly, a cell membrane can block the movement of ions from one side to the other, storing potential energy that is released if a transmembrane channel is opened. An electron orbiting a nucleus also possesses potential energy. This potential energy is converted to kinetic energy as the electron moves closer to the nucleus. We know from common experience that we obtain energy we need from the food we eat. How does food contain energy? Organic molecules such as carbohydrates, fats, and proteins contain chemical energy, which is a form of potential energy. Organic molecules contain many carbon-carbon bonds and carbon-hydrogen bonds. In these covalent bonds, the electrons are far from the two nuclei, so organic molecules store a large amount of potential energy due to their chemical structure.
All right. So now let's take a look at the molecule that we've been discussing for quite a while, that we say has so much, plays such a big role in the storage of energy for our organisms. And that is ATP. So ATP stands for adenosine triphosphate. And if you look at the molecule here, you might think, haven't I seen that before? And in fact, you probably have, because it looks very, very similar to the DNA structures that we've seen before. In fact, adenosine is right here. Adenosine has the ribose sugar and adenine, which is a base that's also used in DNA. Then we have adenosine monophosphate. So that would be this part plus this first phosphate group. You see this is a phosphate group. It has a P and it has 3 oxygens, or 4, depending on how you count it at some point. Well, this one has 4, the others have 3, for example. Adenosine diphosphate would be from here on the whole part. And then adenosine triphosphate, or ATP, contains all 3 phosphate groups. And the bonds linking these phosphate groups have high potential energy. So the bonds in here, the electrons that are stored in there, just like you just saw on that previous little movie from the book, that contains energy, and when those are broken, that energy is liberated, and that energy, when it's liberated, can be used for forming other bonds or producing other kinds of work, performing other kinds of work. So we will just discuss that, and I will show you in just a little bit. But we should remember ATP is the most common energy currency in cellular work. So this molecule is broken down and made, and broken down and made, and that is where energy can be transferred from one thing to another. It's not the only one that can do that, but it's the most common one. ATP can be produced, stored, and used when needed. That's why it's often referred to as a currency, right? Just like a dollar bill, you can use it for this, you can use it for that, you can earn it, you can spend it. And most energy is stored in the chemical bonds between the phosphate groups. So this is where this energy is stored. And you can liberate some energy just by picking off one phosphate group or two phosphate groups or all three. Very frequently what happens is that only this phosphate group is picked off, the energy is used, and then ADP, adenosine diphosphate, is left over, and is then recharged by adding another phosphate group later on. Okay, so this figure shows that what I just basically mentioned, that most energy is stored in the chemical bonds between the phosphate groups in this particular area here. And then when you add water, for example, and you break this bond, then energy comes out of this bond, becomes free energy, and you can have ADP and inorganic phosphate, or PI. So this is often referred to as PI, ADP and PI. So this formula down here, ATP plus water becomes ADP plus PI plus energy, is one that you should be familiar with. Okay, so how can energy be stored in chemical bonds, right? So, I've said that a few times now. Let's spend a little bit of time trying to figure out how energy can be, in a bond. So I broke this down into three different aspects of this statement of energy can be being stored in chemical bonds. So I say here, electrons forming chemical bonds can be moved between reacting molecules. So they can be moved around because they can change their allegiance, if you will. So they can be in between this oxygen and this carbon, but this bond can break and reform in a different way. And after a reaction, carbon may now have only a single bond with oxygen when it used to have a double bond. And that other bond, the electrons there, were now used to create a new bond between this oxygen from a water molecule. And then likewise, this oxygen here can take the rest of these electrons and form a new bond with the remaining hydrogen or proton, and can form a new bond. So you can see that there are electrons that can move around, they can break and form new bonds, and that is how energy can switch around, can move around between and within molecules. So this particular example here is carbon dioxide, water, forming a very important substance that we will discuss more over time that's called carbonic acid. It's often also found as a buffer in cells, but also in lakes or rivers and in the environment. So carbonic acid is important. It's an important compound to know about. All right, so chemical reactions. Let's take a look at how chemical reactions form and break and produce
Chemical
oxygen.
reactions all involve the conversion of reactants to products, and while the atoms maintain their identities, what changes is the arrangement of bonds that connect them. Let's look at the chemical reaction in which carbon dioxide reacts with water to produce carbonic acid. In carbon dioxide, a single carbon atom is covalently bound to two oxygen atoms with double bonds. In water, a single oxygen atom is covalently bound to two hydrogen atoms with single bonds. During the chemical reaction, one of the double bonds linking the carbon atom and oxygen atom in CO2, and one of the single bonds linking the oxygen atom and hydrogen atom in H2O are broken, and two new covalent bonds are formed. The reaction is reversible, meaning it can proceed in the opposite direction as well. In each case, the carbon, oxygen, and hydrogen atoms remain unchanged, but some bonds are broken and new ones are formed.
Okay, so that's basically, chemical reactions, but with respect to how electrons and the movement of electrons between bonds and between atoms within these compounds are carrying their energy with them. So when we talk about reactions, there are reactions that occur easily and fast and almost spontaneously, and others that require a lot of energy in order to proceed. And there are ways how you can describe those different types of reactions. And since that is something that we often do in biology, we need to spend a little bit of time looking at these energetic reactions. So there are two types of reactions that can occur, those that are costing energy and those that are liberating energy. And if we think about ATP as one that can liberate energy, carry energy and then liberate energy, then that liberated energy can be used to produce a reaction that normally would not occur without the input of energy. So here we have so ATP is an example basically of how that energy can be used for these types of reactions. So let's look at this graph here first. So on the y-axis we have free energy, the amount of what's called free energy. And then on the x-axis we have the course of the reaction. So we're starting here with reactants, molecules that are going to react with each other over this time, the course of the reaction. And then at the end of the reaction, we have products that have formed. So just like in the example before where you had carbon dioxide and water, those would have been the reactants. And then the carbonic acid would be the product. So when you have an energy costing reaction, where you put energy in over time, then the amount of energy that is inherent in these molecules after they're produced are put in, and that is called an endergonic reaction. So an endergonic reaction is an energy costing reaction. It doesn't go on spontaneously. You have to give it energy. In a chemical lab, that might be through the input of heat, or light could be one way to get energy. But it could also be the energy that comes through the breaking of a different compound, such as ATP. And then we have exergonic reactions. Those are the ones that are liberating energy. And so there the reactants contain more energy at the beginning. And after they have reacted with each other, their products contain less energy. So the amount of energy that is required for the reaction to occur in an endergonic reaction, that is given as plus delta G meaning the change in free energy, that's the free energy here, and the change in free energy, that's the change from here to there. And if it's positive, if you put it in, if you have to add some, then it's plus delta G. And if energy becomes available, then that energy comes out and is now no longer in the products. And so therefore the overall products themselves contain less potential energy, or less free energy. So in the example of the balls bouncing down the stairs, you would have more energy at the beginning, less energy at the end. Okay, so I put these little words there, these mnemonics, in case they help you. Energy enters the system in an endergonic reaction, and energy exits the system in an exergonic reaction. So maybe that's a way to remember that. And so remember this rule of thumb that a chemical reaction can proceed when their overall free energy is reduced. In other words, when delta G is negative. So when delta G is negative, then overall free energy is reduced from here to here. Then a reaction can occur without input of energy from the outside. Okay, so let's take a look here at this little video showing us more about activation energy.
In which old bonds are breaking and new ones are forming.
We're starting in the middle.
Chemical reactions involve the conversion of reactants to products. In a chemical reaction, some chemical bonds are broken and others are formed. In some chemical reactions, the chemical energy of the products is higher than the chemical energy of the reactants. In other chemical reactions, the chemical energy of the reactants is higher than the chemical energy of the products. Regardless of the energy levels of the reactants and products, all chemical reactions proceed through a transition state that has a large amount of energy. The transition state is an intermediate, highly unstable stage between reactants and products in which old bonds are breaking and new ones are forming. The difference in energy between the transition state and the reactants is called the activation energy. It represents an energy barrier. All chemical reactions, whether they consume energy or release energy, require an initial input of energy to overcome this energy barrier. In chemical reactions that take place in a laboratory, this input of energy may take the form of heat. In a cell, however, the energy barrier is reduced by chemical catalysts called enzymes. The rate of a chemical reaction is inversely related to the height of the energy barrier. The higher the energy barrier, the slower the reaction, and the lower the barrier, the faster the reaction. Enzymes therefore accelerate chemical reactions by reducing the activation energy of the reaction.
Okay, so that showed you some of these reactions. It also went a little further and talked about how to lower this activation energy and what the activation energy is. We'll get to that in just a second. I did want to show you here for these energy costing and energy liberating examples a few products that are exergonic versus endergonic, and that we just know, not from cells, from cellular biology, but just from our daily life. So hand warmers, for example, or these light sticks, they give off energy, right, in the form of heat or in the form of light. Those are exergonic reactions. And an endergonic reaction, just such as this instant cold compress, so something that you mix up, you break, and then it becomes really cold, so you can put it on an injured ankle or something like that. That becomes cold because that energy that is performed inside this pack requires energy from the environment, and so therefore it takes the heat out of the air, and therefore the air around it, or the plastic, or the entire substance here, becomes colder. That's how cooling occurs, because the energy is required for that reaction to occur. So there are different substances in here, they're separate, and then you break them, you break the inner little bags, and then they mix together, the reaction starts, and then when the reaction occurs, it just sucks the energy out of the environment, and that's what allows the cooling effect to occur. So that's an endergonic reaction example. Okay, so but coming back to the question of how energy can be stored in chemical bonds, there is the fact that energy costing reactions and energy liberating reactions can also be coupled. So if we have the endergonic and the exergonic reaction, then energy comes out of one, is required for the other. And so wouldn't it be great if that energy could simply go over from one to the next reaction? And that is in some cases the case if you have compounds that can just be close enough together that the energy can be transferred directly. So let me show you what I mean here. So we have an example where ATP and water produce ADP and PI, right? That is what I said earlier was one of those formulas you might want to remember. And then when glucose is broken down, so sugar, when glucose is broken down, the first step that is required for that breakdown, we'll talk about all the other steps as well a little bit later on in the class, is it needs to be activated. It needs to have an activation by an addition of a phosphate. And that phosphate is the addition of this phosphate, of a PI, inorganic phosphate, requires energy. And so then in order to produce the glucose phosphate from glucose and phosphate, you have to stick in some energy. And so if you couple these two reactions, if you have the first one proceed at the same time as the second time in proximity, then the exergonic reaction, which liberates 7.3 kilocalories per mole, so per number of molecules, and the second substance, the second reaction that requires 3.3 kilocalories of energy to put in per mole, per number of substances, that's an endergonic reaction. So that together can produce this reaction. Glucose plus ATP becomes glucose 6-phosphate plus ADP, because the energy here is, the liberated energy is greater than the one that's required. And so in the coupled reaction, there's a net output of energy of -4 kilocalories. And so this reaction can occur even though It costs energy because it can be coupled with this exergonic reaction at the same time. And so that's how energy can be moved around from one system to another system in order to change the structure of chemical molecules. But there's something important that the little video, the last little video that I showed you, also talked about, and that is that these reactions, even if they are exergonic reactions, usually require a little bit of energy input. So remember the ball on top of the stairs at the very beginning. It just sits there. It has potential energy, but it's not going to start bouncing down the stairs by itself. So you need to give it a little bit of a impetus, right? So you have to push it a little bit in order for it to just start using its potential energy and transforming it into kinetic energy. And that little bit of extra input of energy to get the reaction started, that's called the activation energy. So you have to activate the system. And if you draw this in a similar graph as the ones that I showed you before, with the course of the reaction here on the x-axis and the amount of free energy of the system on the y-axis, then you can say, well, the reactants, they will go from higher levels to lower levels, and that will liberate delta G, and this will be negative, will be negative, and so therefore energy comes out. But if you look at the this energy for this particular example, it's not a specific example, but it's a particular example here. It requires this much energy. This is the amount of energy for the ball to be pushed, right, in order to start bouncing down. So if it's a very heavy iron ball, you would have to push a little bit harder, right? It requires a lot of energy. If it's just a balloon, it will just, with the slightest bit of energy input, it would start bouncing down. So that is the amount of energy that's required to start the reaction. And then once it's started, it liberates all that energy and more and becomes the products with a lower overall free energy. Now, many reactions, biological reactions in cells, are reactions where basically something is burned. And we say that sort of in casual conversation, oh, I burned a lot of energy today. And we don't really mean there's a flame inside our cells that's burning sugar, right? We mean we use that. But in fact, we do sort of burn the energy because just like a fire requiring oxygen and breaking down substances with that energy that is fueling the fire, and the fire being the heat that comes out of whatever you're burning. It's very similar, although at a much smaller level, of course, when substances that contain energy, just like a block of wood, for example, that could burn, when a block of sugar or a molecule of sugar in the cell is oxidized, if it's burned, if it's metabolized using oxygen, just like a fire, to produce substances, and in that process releases energy. And so, in order for these reactions to occur, the transition state, the amount of activation energy that you have to put in for a reaction to start, is sometimes so high that it just couldn't normally occur in a biological system. And so there are helper molecules inside the cell that allow this reaction to occur with much less input of energy, of activation energy. And those are called catalysts or enzymes. So a catalyst is just anything that starts a reaction to go a little faster. It could be an inorganic molecule. But in biological systems, enzymes often act as catalysts. So I say here most biological reactions could not occur without the help of enzymes because the activation energy required would be too large without the help of this enzyme. So this reaction might occur in the lab, but you might have to light a match to it. And you can't do that. You don't have that much energy, nor would you want to do that to your cells. So enzymes, the presence of enzymes, which of course are proteins that have a certain structure and therefore can produce this particular function, when they are around, they will reduce this transition state. They will reduce the amount of energy that's required for the activity, and they can speed up the reaction with less input energy. They cannot change their state here. They do not change the state here. They only change this hump, the amount of energy that's required for the reaction to start. So we have talked about enzymes before when we talked about proteins. And so I want to take you back to these proteins, and I want to show you some structures. And now we want to go back to our, our discussion of how the different types of structures that produce proteins, namely the primary, the secondary, tertiary, and quaternary structure of proteins, how they help in the creation of a function of particular proteins to become enzymes that can have a specific function with respect to lowering the activation energy of a reaction. So remember this space-filling model here. So this is a protein. You see that you can sort of see it in three-dimensional space. You can see that it consists of molecules, of course, amino acids. In this polypeptide, you can't see the amino acids because that's the way that it's shown here. But what you can see is its three-dimensional structure quite nicely. You can see that it has little holes here, crevices and bumps. And you can see that there's a particular hole here that fits a certain molecule just perfectly, like a key in a lock. And so that's usually the term that's used to describe the interaction between an enzyme and its three-dimensional structure and the molecule that it interacts with. And the molecule that it interacts with is called its substrate. So enzyme and substrate interactions occur because of the three-dimensionality of this protein, and because of a very specific way that this protein is structured. So see how it fits right in here? That is called the active site of an enzyme, because it is the place where the enzyme, the protein, binds to its substrate and no other substrate, and it cannot bind to something other than H2O2. It can only bind to that. It's very specific for that. And likewise, H2O2 can probably only bind to this particular enzyme that has the lock and key mechanism where those two will fit together. So oftentimes you see in books this schematic model. So that kind of three-dimensionality is now not shown over here, but you see the active site of this protein. So this is our protein, here's our active site, here's our substrate. So that can fit together. Now I want to go back to what I said earlier a little bit. The function of an enzyme is determined by its three-dimensional structure. And the three-dimensional structure, if you remember back to when we talked about how a protein is formed, depends on, of course, first of all, its primary structure, which is the amino acids that are strung together, but then also how these amino acids, like beads on a string, are then folded together. And remember that the folding occurs because there's interaction between and within these chains. So you can have interaction between the backbone of the protein, secondary structure. We talked about pleated sheets, beta sheets, and we talked about alpha helices. And then there's the way how these helices and sheets and the rest of that protein folds on top of each other in little ways to produce these three-dimensional structures. So when that happens, then this folding brings certain specific amino acids close to each other. And these are some examples. So these purple ones in this particular example are required to hold that substrate just right. So they might be polar or non-polar, they might be acidic or basic, and they might just be able, because of their chemical properties, to hold on to a particular substance that itself has certain chemical properties. So because of the three-dimensionality and because of these rest groups and their chemical nature, you can have this interaction between enzyme and substrate. So I wrote this down here for you just to read again. Protein folding occurs in a specific way because of the protein's primary, secondary, tertiary, and quaternary structures. The primary, of course, is the amino acid sequence. The secondary and tertiary allows the folding of the peptide due to its hydrogen bonds and because of the polarity of the rest groups. And then the quaternary structure is the multiple subunits that's forming. Not all proteins have a quaternary structure. Some don't have more than one subunit, so they wouldn't have a quaternary structure. Okay, so enzymes bind to substrates. The reason why they can fit is because of the three-dimensionality of the protein. So when that happens, that a substrate, let's say a food molecule, a sugar molecule, is bound by an enzyme, and that enzyme breaks it into something else and produces a product, maybe it releases energy, or maybe this product is required for subsequent steps. In that case, you can also have an inhibition of this molecule, this enzyme, by other molecules. So why would you want that? Why would an enzyme ever be inhibited? Why would that be useful for an organism? Well, if this enzyme is very busy and it takes all of the substrate that's in the cell and it just keeps chugging through it and producing products, then that costs potentially quite a bit of energy. It also depletes the cell completely of the substrate. And maybe the substrate is used for multiple things. Maybe it's not just to produce one product. Regardless, there are ways how the cell can stop the enzyme from doing this job without destroying the enzyme completely, because making that enzyme requires a lot of energy itself and maybe some time. You have to translate, you have to transcribe the gene, you have to translate the mRNA into protein. The protein has to be folded. So that can be prevented, that whole breakdown and completely rebuilding from scratch, by simply turning off that enzyme for a little while. And that happens with an inhibitor. So an inhibitor can bind to an enzyme and render it inactive temporarily in two ways. Either it binds to the active site. So it says here, in this case, the inhibitor binds to the active site of the enzyme, competing with the substrate and reducing the rate of reaction. So now it's competing with the substrate, and the substrate can no longer bind there, and so therefore it's no longer reacting, because the inhibitor binds more tightly, or at least if it's binding as tightly as the substrate, then at a 50-50 ratio, where it's just random whether one or the other binds, even that would reduce the amount of substrate being turned over into product. The other way is that an inhibitor can bind to a different site in the enzyme. So this is called here the inhibitor site. And if it binds to a different site and changes the shape, so it binds here, and then that binding maybe pulls the chain a little bit closer together, so that at a different place in the active site of the protein, that chain now becomes wider, and so that active site is no longer really well suited for holding the substrate. Well, that is also a way how you can get inhibition of enzymatic reactions. So either through blocking the active site, or through binding to a separate site, an inhibitor site. So why is this important? It's important because certain reactions aren't always needed, right? So this reaction is great, but it's not always needed. So therefore, when it's not needed and it can be temporarily turned off, that would save energy. It's also a great way for the cell to know that it might have a lot of this product if that product itself could bind to the enzyme and inhibit it to produce what's called a feedback reaction. And that does happen. So this inhibitor can either bind one way or the other, but the inhibitor can either be a completely different molecule, just like in this case it's red as opposed to yellow and blue, but it could be, and it is the case in some specific cases, that the product that is made in this reaction itself is an inhibitor. So if you have a lot of product, then maybe the cell doesn't need to make any more of that. So producing a ton of that will then at some point slow down the reaction with which this product itself is made. And that is what I mean here with number 2. If the reaction's product is inhibitory, it can provide feedback. So it's feeding back to the system. So, and why is that important? Where is that important? Can we have an example here? Yes, we can. So in the example of the production of isoleucine, which is a amino acid, which is made from another amino acid that's called threonine, through a number of steps that require a number of enzymes. The example threonine dehydratase, which starts reshaping threonine a little bit through all of these steps to produce isoleucine in the end. Well, when isoleucine is around, that product itself is a inhibitor of threonine dehydratase. And so that is called an allosteric inhibition. So allosteric meaning someplace else in the enzyme, on the enzyme binding, creating a feedback so that the enzyme now no longer works. And so that's why I say here, once the cell has enough isoleucine produced, continued production is turned off by enzyme inhibition. And that saves energy and raw materials, right? So threonine is also needed. So no longer will all the threonine be produced, be used to produce isoleucine. Now the cell has some threonine itself left that is not being siphoned off because this reaction here has been shut off temporarily through this inhibition, this feedback inhibition. All right, let's just sum up what we discussed today. We talked about the fact that chemical bonds can store and release energy. I said that ATP is the universal energy storage molecule in living cells. Energy costing reactions can be coupled with energy releasing reactions, such as the breakdown of ATP. We talked about the fact that enzymes reduce activation energy and allow reactions to occur in living systems without the high input of energy. We talked about enzymes containing active sites that are specific for a substrate. We looked at the 3D structure of an enzyme and how that determines its function. I talked about the key and lock system. And then we said that enzymes can be inhibited, for example, by their own product in a feedback loop. And so from here, what we're going to do is we're going to take a look in the next lecture how cells break down molecules that are energy rich, so for example sugar, and how enzymes are involved in the breakdown and how ATP is produced or used to activate parts of that system, and how in the end a lot of ATP is gained through a process called oxidative phosphorylation. So those things are coming up in the next two lectures where we're looking at the breakdown of sugars to the production of ATP. And I'll see you then.